Hello everyone, a very warm greeting to one and all. My name is Sri Gauri Krishnakumar and I would like to welcome you all for session 1 of omicsLogic Genomics program. So what is this program all about? Advances in genomics are contributing to the development of more effective personalized approaches to the prevention and treatment of infectious diseases. From tracking pathogen outbreaks to fighting infectious diseases, bioinformatics aided genomic sequencing data analysis continues to be a promising field of scientific research. As a part of session 1, Genomic Data Analysis, we will be covering the topics on next generation sequencing data, omics data, molecular determinants of a phenotype, overview of mutations and variations, course structure, expectations, and intended outcomes. Topics include applications of bioinformatics, introduction to bioinformatics course, examples in biomedical research, pharma R&D, and agriculture. So before we begin with today's session, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The omicsLogic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. This training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. So how do omicsLogic resources help advance bioinformatics teaching? omicsLogic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, omicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This program comes with an access to the omicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, and example projects of different fields which you could learn from and replicate for your research work. Users will be given directions to access the tBioInfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections on this multi-omics platform. So first of all, let's begin by signing up on the omicsLogic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, or ID, or even Twitter. A brief bio about yourself that will consist of educational background and research interest. The mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the portal. I'm sharing the link to the portal in the chat box so the users can sign up on the portal. All right, so please click on the link and I'll walk through the process behind signing up on the portal. And for users who do not have an existing account on the portal, please click on the Create an Option account that you see here. So here you will be asked to enter your name, email ID, and your password. You also have the option to sign up using any of your social media accounts, which could be your Google, Facebook, Apple account, GitHub, or even Twitter. With that, I'll pause for a couple of minutes for the participants to sign up. Once you have signed up, please put a yes in the chat box. And if you're facing any technical issues for signing up on the portal, you can share that in the chat as well. And our team will be happy to help you. So I'm pausing for a couple of moments. And once you sign up, please leave a yes in the chat box. I'm monitoring the responses in the chat box. All right, we have one yes. I'm waiting for, okay, 2 yes. I'm waiting for rest of the participants. Okay, great. So, all right, so since I already have an account on the portal, I'm going to click on the login now option and sign in using my existing account. So once you sign in, you are able to view the various courses and example projects under the courses tab. So since most of the participants are interested in genomics, you can browse through the various example projects and courseworks that are available on genomics. All right, so now the most important step that I discussed previously was setting up the profile on the portal. So for that, please click on the tab that says welcome back with your email ID. So how do we edit the features that I mentioned before? For that, click on the icon that says profile. And here you can enter the necessary details such as upload your profile image, enter your bio, your name, link to any of your social media handles. So in my case, I've linked my LinkedIn and my org ID. So under the activity tab, the mentor and the community manager will be able to track your learning progress. And once you start completing any courses, it will appear under the courses tab. So in my case, I'm currently completing Python course 2. And if you're completing any projects and you're uploading, it will appear under the projects tab. And once you enroll for any of the program, for example, the genomics program, it will appear under the programs tab. And finally, once you complete a course, the certificates will appear under the certificates tab. All right, so if any of the participants have any queries so far, please put them in the chat and I'll be able to guide you. Otherwise, now I'd like to pass on the stage to Dr. Raghav to start with session 1 of Genomics program. Over to you, sir.
Thank you, Sri Gauri. Let's find ways to share my screen while you're all busy signing up for the portal. Can you see my screen?
Yes, sir.
And hear me clearly. Okay, that's great. So I am actually monitoring the chat. So if you have any queries, please immediately type in the chat box. And if that is appropriate, we will discuss it immediately, or if not, then I will address those questions after the session is over or towards the end of the session. So welcome to the first session of Genomics Data Analysis, where we are going to discuss how we can process, analyze, and interpret genomics data in general. So in today's session, we will focus on the code, DNA code itself, and we will focus on how to interpret the variation. That is the main focus of analyzing these genomics or these DNA code themselves, and what are the different ways in which these variations can tell us or can help us understand the biology behind the subjects or biology behind the study samples that we are investigating, that we are studying. So let's start by going through a couple of slides of review. So if you already know this, you can speed it up, or I'll follow it along. So let's start reviewing the fundamentals of genomics. Genomics is all about DNA, so we know that. So let's also review what we know about DNA code and what are the answers that we can find inside the DNA code. And we also know that a DNA code is they comprises of huge sequences of nucleotides that are stored in this DNA, and how do we make sense of these huge number of sequences, and what do we do to make sense of them? What do we mean by making sense of this DNA code, right? So if we attribute the DNA code or genomics data to a book, then the code can be thought as to be organized from letters that will form sequences, sentences, and the sentences will form paragraphs, and paragraphs are organized into chapters. And understanding and knowing how to read this code can help us decipher the full story about who we are, can help us understand to look into our history, can also help us understand how we as an organism or as a being are connected to other living things that are around us. So when we talk about genomics, the first thing that comes to our mind is genes or codes. These codes or genes are the ones that are passed from cell to cell, and we have approximately 3.2 billion letters in total, and these encodes to around 22,000 proteins. And these genes are made up of biochemical entity called DNA, which is a short form for deoxyribose nucleic acid. This DNA molecule is a double helix, that is, two long thin strands that are twisted around each other like a spiral staircase. So in this DNA, the backbone of each strand is made up of sugar and phosphate, and these two strands will interact with each other through nucleotide bases. That is what we sequence. So as we spoke about that, this DNA is made up of nucleotides. So the nucleotides themselves are made up of three important components: a sugar ring that is called deoxyribose, a nitrogen-containing base, and a phosphate group. These nucleotides are the ones that are joined to one another in a chain by covalent bonds that form between the sugar molecule of one nucleotide and the phosphate group of the next. This results in an alternate sugar-phosphate backbone, and the nucleotides of the two complementary strands are connected together, and this connection that connects two nucleotides of the complementary strand is called base pairing or base pairs. The base pairing connects these two strands of DNA, and this connection gives to the characteristics double helix form of the DNA. So in this example, in this structure, we can see that the adenine is always bound to thymine, and the guanine is always bound to cytosine. And they form the four bases of the nucleotide A, T, G, and C. So that's a very short introduction about the DNA, and its basics. So let's look into various ways where we can spot the DNA variation, and how can those be helpful for us in understanding the biology behind it. So we spoke about this, and what do we mean by studying the DNA variation? When we discuss about studying DNA variation, we always want to compare the sequences that we have in the current sample that we are that to the reference sequence, and to identify what are the places in which the sequences or the bases vary or bases differ generally. So that this differences in the bases, or this differences in the nucleotides, or this differences in the in this DNA code can give rise to unwanted effects, which we call as mutations, or typically they are termed as DNA variants. Right? When these variations affect the end product, which could be a protein coded by the specific gene, then we will call this variance as mutation. We will speak about this a little bit later. And depending upon what type of variant that we are getting, the end product, the protein can be altered in their sequence, and that alteration can affect the functionality of the protein, and hence we are ending up with a pathogenic variant that is termed as mutation. So the reference sequence, where do they come from? So this reference sequence actually comes from the first human genome projects, which is what enabled us to compare any given DNA sequence from a human to a common reference. This common reference is what is compiled from multiple individuals. And this project is a 13-year-long project which started at 1990, and we all know about what this project gave us till date. So the primary goals of this Human Genome Project was first to determine the DNA sequence and the location of the estimated genes. The estimated number of genes at that time was around 100,000. But now we have filtered that, and we have come to a consensus that there are around 21,000 to 22,000 genes in our whole genome, and all the other sequences are present to regulate the translation of these genes into proteins. Most of the other sequences. So this is the reference sequence to which we will compare any new sequence that we will generate, and identify the differences in the sequences and try to characterize the differences in the sequences, which in a nutshell can be termed as genomics data analysis. So this resulted, or with the advancement in the next generation sequencing technology, we have now actually obtained, or there is an ever increasing number of genomes that are sequenced, and the cost of the sequences have drastically decreased, and this gives rise to more number of sequences being generated, and we in the end are ending up with more and more data that has been deposited to public repositories like NCBI and EBI, and we can make use of all these public repositories to make meaningful data analysis and understand the genomic variations that are present in the samples in general. So this plot indicates that the decrease in the cost of genomics technology generally followed Moore's law until one point, and after that it drastically reduces, and which can actually give you, or which can actually give you an indication of how the technology has genomics technology has improved leaps and bounds. So what do I mean by understanding the differences? We already spoke about that. A little bit more background for this. Efforts such as the one described in the previous slide, variety of different numerous different studies that will try to understand variances or variations that in the genomics code compared to the reference code, it can now, for example, detect harmless and also harmful variances present in the sequences that we are actually sequencing. Just like any code, or just like any sequence of letters, we can expect the DNA to contain mistakes, and these mistakes actually occur as a result of copying the DNA source too many times, and also these mistakes can also occur as a result of various stresses that the cells will undergo, and these stresses and the result of copying may actually interfere with the copying mechanism of the DNA and can introduce mistakes. And these mistakes are the one that we are focused on detecting. And when these mistakes actually occur in important regions like genes that will encode very important proteins, and even inside the genes, important stretches of sequences that result in key changes in the proteins that will end up altering the shape and size of the protein, and it can also end up altering the shape and size of the cell as it is given here, can actually lead to serious issues when that gene is being produced. This is expertly understood in the event of sickle cell anemia, where a normal protein sequence encoded by the gene actually produces red blood cells of a normal morphology, and when one single nucleotide change actually changes the amino acid from glutamate to valine, and that results in producing red blood cells that shape like sickles and result in a reduced carrying capability of oxygen, and hence it results in anemia. So this is one of the example of mutation or point mutation, one of the examples of mutation that occurs in single nucleotide position. And all the mutations that occurs in single nucleotide positions are actually defined as point mutations. Depending upon what is being changed, there are several uh, types of point mutations that one can characterize. So one among those is substitution mutation. A substitution mutation can cause several consequences to the DNA code, but this substitution mutation implies minimal change that only leads to single nucleotide change, as we actually discussed. So what is changed? One nucleotide that is in the reference sequence is substituted by another nucleotide, and this resulting substitution mutation may or may not end up changing the amino acid sequence that the trinucleotide codon codes for. And there are also other forms of single or point mutation, which could actually involve insertion of a single nucleotide into a position in the altered sample sequence compared to the reference sequence. This insertion, unlike the substitution mutation, may end up changing the reading frame and that encodes a trinucleotide codon, and hence from the point where the insertion is observed, the resulting protein sequence may be completely different from the one that is present in the wild type or the reference genome. And this may result in the protein that may not function properly or that may prematurely end. And contrary or complementary to the insertion mutation, we also have deletion mutation, where one nucleotide from the sample sequence compared to the reference sequence is deleted. Just as in insertion mutation, this deletion mutation will actually change the reading frame of the trinucleotide codon, which produces amino acid sequences, and hence it results in the protein sequences that is completely different from the wild type variant. And this may also, or this is also most likely expected to change the functionality of the protein and cause serious defects in the cell's functioning and general cellular molecular function. So unlike substitution mutation, insertion or deletion may actually contain more than one number of mutation, one number of nucleotide, and again in those instances the resulting protein may completely differ. And there is another form of variation or another form of mutation, that is duplication, which consists of a piece of DNA that is abnormally copied more than one or more times in different places or in an adjacent location. This type of mutation again may alter the functionality of the resulting protein, and some of the examples of these mutation we will discuss in detail later. So depending, we spoke about point mutation, and focusing on the types of mutation from the point of view of the amino acid sequence that these mutation encodes for, we can again categorize these mutation into missense, nonsense, and frame shift mutation. So a missense mutation is characterized by the change of point mutation at the nucleotide level that result in the changes in the amino acid sequences, right? Sometimes this single amino acid change that occurs in the protein may occur at a structural or functionally important location and may end up in greatly changing the protein's function. So in that event, as we spoke about, as we discussed, the end result may actually completely change the function of the protein and hence affect the proper functioning of the cell completely different. So a nonsense mutation is actually a change in the nucleotide base, prematurely stops the protein production by introducing a stop codon at that specific position. So this definitely changes not only the protein sequence themselves, but it stops the protein sequence production prematurely and results in a shortened length of the proteins themselves. So this can result in the protein folding incorrectly or not folding at all, and hence the functionality of the protein is completely lost. A frame shift mutation is what results in when we have an insertion or a deletion, as we already discussed, and this changes the frame which reads the trinucleotide codon that encodes for different amino acid sequences. And hence, since the reading frame is changed by the insertion of this one adenine at this position, now whatever is encoded after that insertion position or after that specific point mutation is completely changed, and this again will result in a protein sequence that is different from the wild type, and this differed or this mutated protein sequence may again result in incorrect folding or result in a completely different function than what the original protein is intended for. All these will actually affect the cell's functionality to a larger extent, and depending upon what is where this changes occur in the morphology of the tissues and how they actually occur and how they actually affect the cell and tissues and hence the organism, they can be characterized into genetic variation compared to mutation. So generally, all the changes that we observe in our sample compared to the reference sequences are termed as genetic variations or genetic variants. So the genomics data analysis focuses on identifying these list of genetic variations and characterizing, annotating them into potentially harmful and potentially not very harmful, and that the one that can be ignored by just characterizing them to a different level. So for example, if you see the morphology of this, for example, in this example, we have a wild type form, which in its natural form, without any mutation, has this specific morphology. And there could be some variations in their genetic gene sequences that can change, for example, the eye color, that can give a couple of more modifications to the morphology. But if these mutations are, no, not mutations, if these variations are not going to change the functionality, are not going to affect the survivability of that specific form or organism, they are not termed as mutation. They are generally considered as genetic variants or polymorphism, etc. But variants that results in loss of function, that results in improper function, that can cause diseases or that can cause genetic defects are termed as pathogenic, and hence they are called as pathogenic mutations. Sometimes in several of the publications, the word mutation and variants are actually used interchangeably, but generally when you refer to a mutation, we always refer to the pathogenic version of the variant. And when you refer to variants, we either refer to changes in the nucleotide sequences that are not characterized as of now or as of at the moment of data analysis, or they are characterized to be inconsequential to the functionality of the tissue or organism. So that is the difference in their terminology that we have to keep our mind to. So as we spoke about, how do we identify these genomic variations, right? These genomic variations can be identified by subjecting the samples into high throughput sequencing or next generation sequencing, which you will learn a lot in the upcoming sessions. And when we map all these reads to specific position in the genome or reference genome, then we can identify the places or the positions in which that specific nucleotide is changed, or that specific nucleotide is converted into a variant or a mutant form. When we speak about single nucleotide variant, excuse me, my is frozen. Let me fix that. Okay, let me reshare again.
Hello, Dr. Raghav. So we cannot see your screen.
Yeah, I'm resharing again. It completely froze.
Okay, now it's visible.
Yeah, can you see my screen?
Yeah.
So thank you very much. So when we speak about the genomic variation that we are trying to characterize, let's refresh our memory that what we have been discussing and summarize into 4 or 5 points. So single nucleotide variant, or SNV in short, can be characterized as a single nucleotide change that may or may not be present in whole or most of the population. Basically, this means any change that we observe in our sample from the reference genome is can be termed as SNV, or single nucleotide variation. While another term that we always hear or that we always discuss is single nucleotide polymorphism. For example, when we have samples that typically consist or comprises of more than 1% of the population, or more than, for example, not more than 1% of the population, and when we observe this specific variant in more than 1% of the population, and that variant is considered to be single nucleotide polymorphism. So when we refer to this 1%, that refers to the whole population set of that specific group of sample that it refers to, not the sample themselves. So now you can for example, appreciate the amount of sequencing that has to go into estimating the 1% change of variant that is present in 1% of the whole of the population, to term them as SNP. For example, the SNPs that are characterized for the ethnic group and different races like Caucasians, different races like Asians and Europeans, they actually involved in sequencing more than 1%, or approximating to sequencing more than 1% of the population, to identify these single nucleotide polymorphisms for that specific group of samples or population. So any variant that we find to occur above the level of technical error can be termed as single nucleotide variant, and any single nucleotide variant that is prevalent in approximately 1% of the population can be termed as single nucleotide polymorphism. And another type of variation that we often refer to is a copy number variation, where a gene is encoded in multiple copies in our genome, but the number of the copies varies between different samples and between different sequences. And when this number of copies actually varies significantly between the reference sequences and our sample sequences, then that may also result in the changed level of gene expression, and hence may actually result in altered phenotypical condition of the cell or tissue or organism in general. So all these changes that are characterized at the level of the genes or genome are termed to as genotype, and many of these genotype can actually end up in specific phenotype that can be harmful, or that can be harmless, or that can be potentially ignorant, or potentially beneficial in some aspect. Right? For example, in this specific gene, one of the examples of harmful mutation or harmful genotype is the one that involves PAX genes. In this specific example, PAX6 is a gene that will encode for PAX6 protein, and it has been characterized and recognized that this PAX family of genes is necessary for the development of eye in almost all of the organisms. And surprisingly, the development of eye in many of the organisms follow similar developmental path. So we can follow the progression of this gene and their defects in many different model organisms. And experiments in such organisms like mouse or mice samples demonstrate that deficiency in PAX6 will lead to not only in decrease in the brain structure of normalcy or normality that leads to autism. It can also lack complete formation of iris, or it can result in cornea, and that will end up causing blindness in the blindness in the variant or in the phenotype, blindness as part of the phenotype. This is one of the examples of harmful mutation. There are a couple of examples of potentially harmless variants. For example, some mutation in specific gene can cause long hair growth in eyebrows or in eyelashes. They can be potentially a little bit difficult to deal with, but they are not harmful in any case. One more example of, for example, potentially not very harmful, but advantageous mutation variant is the SNP that is linked to or linked with the development of blue eyes for the Caucasian is the variant that is linked to the blue eye of Caucasian is actually an SNP that is characterized for European and US race people. So these are all different ways and different forms of variants that can or not that may or may not end up in causing difference in the phenotypical versions of the samples. So such analysis to understand and characterize these variants can be performed at many different levels to investigate the functions of variety of different genes in multiple different research areas. Right? These research areas can include drug metabolism by studying the enzyme function that is key in the catalysis and the key in the functionality of specific metabolite. And this can also end up trying to characterize, we can also end up trying to characterize mutations in specific genes that can cause tumor. In that case, genes that are linked with tumor progression and tumorigenesis. And we already spoke about sickle cell anemia. This in case, this in this example is caused by the effects of germline mutations that are inherited from our parents. So furthermore, one can also use genomics to explore pathogens and to understand important properties of their pathogens as a result of their or as a function of their genomic variants, to study virulence, to study disease transmission, and to study drug resistance, etc., etc. Many of these examples will become clear as we review several projects, project and exemplar projects as we go along in the sessions, as we go along in the program. And some of this we can also discuss in the upcoming slides in today's session. So if you have any questions, please let us know in the chat box. We will discuss that before going to specific examples that will help us understanding the genomic variance analysis. Okay, if there are no other questions, let's proceed. So until now, we have tried to understand the concepts and the types of variants and mutations and how to, for example, characterize them. So let's understand why and how these mutations. Yes, Akshita, this will be available. So let's understand how these mutations and variants play very important role in the progression or development of cancer, or better, can we say, how can the mutations will actually help in tumor formation? So we already know that there are various important genes that play key roles in cell cycle and regulation of cell growth and etc. So any disturbances in these genes will actually result in dysregulation of cell growth and eventually enhancing the development of tumor. So if we focus on those genes and focus on variants and variations on those genes which could result in tumor development, we can actually discuss a couple of examples. For example, these genes are actually generally termed as tumor suppressor genes because usually these genes regulate the cell growth, and if they for example, if they encounter any defects in the cell growth, they end up inducing apoptosis and hence saving the saving the eventual progression or eventual progression of the tumor and development of tumor and progression of the tumor in these samples. But if the genes, if these tumor suppressor genes get mutated and, for example, get inactivated, it will or the mutated version may end up altering the normal function of these genes, then the regulation will be affected, and eventually that could result in uncontrolled growth of these cells. So some of the examples of tumor suppressor genes that you can easily relate and you can actually find everywhere in the biomedical research area is the TP53 that we all know about, and P10 or some of them. So these genes that are responsible for cell growth and potential mutation that is caused in these genes may result in promoting, for example, promoting tumor are termed as oncogenes. And there are also some of the genes that are responsible for cell growth, and if any mutation in these genes that are responsible for cell growth can be related to promoting or accelerating or actually amplifying their normal function. That was the word that I was looking for, amplifying the normal function. And these genes at their normal level that promote cell growth, but at their amplified level can promote abnormal growth are termed as oncogenes. And any mutation that results in uncontrolled cell growth will again result in tumorigenesis. And we can actually find some of the examples of these oncogenes that are HER2, which is epidermal growth factor receptor, human epidermal growth factor receptor, HER2, that is one of the oncogene, and some mutations in these genes will actually result in these receptor binding to a lot of growth factors and promoting abnormal growth of the cell. And there is also another oncogene, which is epidermal growth factor themselves, right? EGFR, epidermal growth factor receptor themselves. And this again will bind to many growth factor molecules and promote tumor progression and tumorigenesis in general. And there are also other forms of genes and other genes which are involved in DNA repair mechanisms, for example, and any mutations in these DNA repair mechanism involving genes may also end up changing the characteristics of the cells to a malignant level. So some of these mutations can be acquired, which is the most common cause of the cancer. They can be acquired due to stress. They can be acquired due to many times copying the DNA code to several generations, and they can also be acquired due to random chances, right? And some of these mutations can also be a result of hereditary cause, or these are all termed as germline mutations, which are less common. And the genes that are famous for germline mutations are the BRCA genes. So if specific mutations in the BRCA1 and BRCA2 genes get propagated from the parents to daughters, or from the parents to cells, from the parents to progeny, then that will result in most likely or result in a higher risk of developing breast cancer and ovarian cancers, etc. So some of the mutations that are acquired, for example, may be concentrated in several different portions of the gene, and these are all somatic mutations or acquired mutations. And those mutations which are known to cause tumorigenesis or tumor progression or cause cancer are termed as driver mutations. And those mutations which are known to be associated with the presence of cancer, but their clear relationship with the cancer development, if that is not known, then those mutations are called passenger mutations. And when one discusses, or when one focus on one specific gene, for example, TP53 gene, which is very well known for the protein that is tumor protein TP53. And this protein, as we already discussed, acts as a tumor suppressor and regulates the cell division by keeping the growth and cell division under control. So the mutations that are present in TP53 are of numerous type and also numerous variety. And you can actually check, right? If I did not butcher your name, you can go to repositories like Cosmic and mainly Cosmic to study potentially pathogenic somatic mutations related to one specific gene. So if you type TP53 gene in Cosmic, then it will end up listing all the known variants and mutation that is known to cause that is known to be associated with the tumor development themselves. So there are also other forms of annotation in the Cosmic database, which will annotate the mutations to be potentially a driver or potentially passenger or unknown causes, etc. So this TP53 gene themselves, it is implicated in many cancers, not only in specific type of cancer, many cancers, and it helps, as we already discussed, it induces apoptosis, and when the protein actually detects some abnormality in the genes and several of the mutations that is present in the specific regions that bind and recognize DNA code can lead to be promoting tumorigenesis by not controlling the replication of the cells with damaged DNA. So this protein will bind to DNA and will identify incorrectly translated or replicated bases, and when you observe several mutations in the binding site of these genes, that will actually that are actually usually associated with tumor promoting driver genes. And there are also other variations or other variants and other mutations that are generally observed in the structure of the TP53 genes protein, which may not be directly related to their functionality, but one has to say, or one has to keep an open mind that we have not completely understood the effect of those mutations until now, even though we have understood many different mutations and their involvement in tumor progression and tumorigenesis for TP53 gene in specific. So the relationship between disease-causing mutation is not limited to cancer. In fact, this relationship and prevention has developed a lot, has been, for example, worked upon in a lot of other diseases compared to cancer. Like one of the very well-known story is about cystic fibrosis, right? Cystic fibrosis is as we know, can be understood as it causes chronic lung infections by the changes in the mucus secretion. So it is usually or generally caused by mutations in one single gene, that is CFTR, cystic fibrosis transmembrane conductance regulator gene. So this mutation can actually result in either no CFTR protein being produced, or some of the proteins that are produced may be ill-formed or malformed, and they cannot perform their key functionality in the cell. And there are, depending upon what happens or depending upon the variety of different variants, there are 5 classes of these CFTR mutations, and they can occur in different stages from the protein synthesis to producing insufficient protein. The most common disease-causing mutation in all these 5 different classes of CFTR is this protein processing or maturation processing protein, where this one specific amino acid, phenylalanine, is actually deleted. And this actually occurs in more than 80% of the patients, and that causes incorrect regulation of the mucus formation, and hence it results in chronic lung disease. There are also other mutations like the one that is in like the one that is in the gating, that involves the gating mechanism, like glycine to aspartate, and which causes complete loss of the protein in the plasma membrane by resulting in the gate being too narrow. So generally, the life expectancy of these cystic fibrosis patients can be improved or can be lengthened when we diagnose these mutations early, and when some of the symptomatic treatment therapeutic regimens are produced. But these are actually results of hereditary mutations, right? Or they are germline mutations, and one can observe the presence of these mutations when we look for these specific mutations from the development stage of the progeny themselves, right? So depending upon where these mutations occur, as we see that there are several advancements in understanding these CFTR in understanding these CFTR mutations has been made, and specific gene modulators for these specific mutations are also developed, and these modulated drugs have the ability to, for example, restore partially or fully the functional expression of these specific CF causing mutations, and they also have been classified into addressing these specific mutation separately or accordingly. So this highlights the importance of studying genomic variants. No, I mean, yeah, that's a great question, Komal. Why only 39 mutations of CFTR is considered for modulated treatment? Why not the other 1,700 mutations? So we can only act upon those mutations for which we can successfully generate or develop modulation. There may be several other mutations, and many of these mutations may remain uncharacterized or may not be reachable themselves. The modulators are also therapeutic drugs that has limited reachability or that has limited functionality. One has to identify, or one has to use only certain targets of these certain target mutations of these CFTR. No? So that is why people are still working on these modulator productions, and CFTR is very actively developing research area. Understanding CFTR mutations is very actively developing research area. And one has to keep saying that these modulators will only partially, may only partially restore the functionality of the CFTR, and several more research has to be successfully developed and successfully performed to generally increase the quality of life of these patients. Until now, we have only succeeded in extending the life expectancy of these patients, and eventually when we end up in improving the quality of the life, then people can actually consider this as a success story. But nonetheless, understanding the mutation at several different molecular level will actually help us generate specific intervention for those specific variants or mutation, and can actually help the help treat patients suffering from these deadly diseases in this condition, right? So mutations classified at, for example, categorized as at single nucleotide positions are not the only cause of disease, not the only disease causing mutation. For example, in the case of, I'm not a professor yet, Komal, thank you. So in the case of Huntington's disease, which is a rare inherited disease, we already know they cause, for example, progressive breakdown of the nerve cells in the brain. And this has a wide impact in person's functional abilities, and it usually results in movement disorders, usually result in cognitive disorders, and may end up resulting in psychiatric disorders in the end. For example, we know that Huntington's disease is an inherited condition where instead of point mutations that we have been discussing so far, a specific gene on the chromosome 4 has an abnormal number of CAG codons. This is an example of duplication mutation that we discussed, or that we just touched upon earlier. So where the CAG codon is actually multiple times reproduced or multiple times duplicated in the HTT or Huntington gene. So this gene encodes a very large Huntington protein, and these additional repeats actually change the structure of the protein completely, and this is the result of progressive movement disorder or dementia, or behavior abnormalities, etc., etc. For example, the Huntington diseases typically emerge in around midlife, 45, 55, or even later at that point. But there are certain cases and there are specific cases where juvenile onset has been observed, and when we can, if we can diagnose the presence of these characteristics motor signs, based on the presence of these characteristic motor signs, then we can look for these mutations in those juvenile onset cases. That is what is being done by the Huntington Consortium in 2015, which published a study of over 1,000 individuals with early and late onset of this disease. And by understanding the whole genome sequencing and understanding the targeted sequencing of specific chromosome 4 sequencing, researchers discovered very important regulatory mechanisms that are differentiated between the groups, for which we observe early disease or early onset, and for another which we observe late onset. And that actually is associated with the chromosome 15, where we have abnormal copy number variations of a specific gene, and that results in the slower development of the of this specific disease progression, and etc., of different mechanism of disease progression. And this chromosome 15, for example, this specific gene in the chromosome 15 that have abnormal number of copy number variation can actually help us identify mechanisms of this disease progression and help us develop therapeutic therapeutic intervention. So now what I was actually trying to say is the chromosome 4 is what is associated with the development of the Huntington disease. But when they perform this targeted sequencing effort in these 1,000 individuals with early onset and late onset, their differential variant analysis resulted in identifying specific regions of chromosome 15 that had higher number of copy number variation or higher number of copy number aberration. And for them, right, those for those who contain this higher number of copy number aberrated chromosome 15, they are associated with the slower development of this Huntington disease. And by understanding this variant in particular, one can identify mechanisms of this disease progression and relate that to the therapeutic intervention by naturally understanding the disease regulation and etc., etc. That is what this study actually helped us understand in specific. So where without any intervention, and natural variation that occurred for a specific group of people acted as a potential regulatory mechanism for a deadly disease, we can exploit to treat the specific early onset of Huntington's disease. So proceeding further, we can actually turn our focus into pathogen genomics data analysis. In this case, we already know tuberculosis is so difficult to treat because they usually attack the lungs and they can spread to other parts of the body like brain, spine, etc. And this is caused by Mycobacterium tuberculosis. Why is this so important? In the 20th century alone, tuberculosis was the leading cause of death in the United States, and today most cases are usually cured with very difficult to follow and very complicated antibiotics regimen, right? But in the last 3 years or 4 years, researchers have actually discovered that this bacteria has become more and more resistant to not one, but several different types of antibiotics. So it turns out that this resistance can be associated with a specific set of mutations in specific genes or their promoters. So understanding which gene and which antibiotic that gene mutation, resistant to which antibiotic that gene is associated to, are very important in understanding the development of resistance in general. So there are also large, for example, part of developing new drugs to address this surge of multi-drug resistant tuberculosis infections themselves. And in addition to this multi-drug resistant tuberculosis, there is an existence of extensively drug resistant tuberculosis, which is resistant to all different types of antibiotics, or most of the different types of antibiotics that it has been tested. So there are multiple studies that focuses on understanding the population level or population percentage distribution of these multiple drug resistant and extensively drug resistant tuberculosis from single infected isolated area. And there are also studies, for example, in as you will know, in some of the example projects that we have, study 22 years old study performed in single patient to understand the development of drug resistance to variety of different drugs that are used to treat that specific patient. So we have more examples, like examples in Ebola virus, where we know that the viral protein VP24 is a nucleocapsid protein that is associated with inhibiting interferon gene expression and interferon mediated antiviral response. So this actually prevents the nuclear import of the signal transducer and the activator of the STAT1, and hence the interferon mediated antiviral response is heavily depleted in the Ebola virus affected infected patients. But several studies on specific strain of Ebola virus that exist with reduced transmissibility and reduced disease pathogenicity or virulence actually identifies specific mutations at residues 142, 146 that are able to, for example, reduce the activity of the VP24 by 90%, right? So these mutations that are naturally occurring were the result, or were the were one of the starting point that ended up helping us develop vaccines for this Ebola virus in this specific epidemic at 2014, 2016. So there are other examples as well. For example, this considers, or this actually discusses the use of understanding the rhizosphere, which is defined as the soil zone around the plant roots, right? And this soil, as we know, is rich in nutrients, and in this region there is an intense interaction that takes place between the plant root tips and plant roots and the microbes that are present in the soil, microbes that are present in the plant roots, right? These microbes inhabiting area are generally termed as rhizobacteria, and they can actually regulate all the known biogeochemical cycles of this specific space, and they can regulate degradation of organic chemicals, and they can actually preserve the components of the soil chemistry. And understanding the mutability or mutation of the, for example, mutability mutability in all the positions of this rhizobacteria genome can actually help us understand the variety and variation that is present in several different rhizobacterial genome in specific. There are a couple of publications that are involved in isolating specific rhizobacteria genome and in a genome-wide association studies, GWAS-like studies in understanding the heterozygosity of this specific rhizobacteria, which we know are very beneficial to maintaining the healthy ingredients of the soil biosphere. People actually characterized mutations that are beneficial to maintaining these interaction at a healthy rate, and some of the mutations that are known to cause depletion in the functionality of the rhizobacterial genome in general. So that is also possible. So this is not only limited to, for example, understanding the bacterial genome that that inhibits or that are inhibited of the inhibited of the soil rhizosphere. One can also use general genomics data analysis in promoting crop yield and in promoting understanding the genetics of agriculture, which precedes the use of genomics in agriculture by several decades. So the efforts in those areas are also heavily heavily advancing, because generally when you see the amount of data that is available for us to compare our samples with are focused more on human genome and disease causing human genes and less on general crop yields and crop production and agriculture in general. So in those aspects, people focus on not comparing the genome with the reference genome. If the reference genome is not present, then they work out, or then they perform GWAS-like studies in trying to sample variety of different genomes and understand the variation that is present at different levels inside the plant sequences, etc. So this is not limited to only plants. This can exist at different levels. Also in metagenomics levels, also it can you will get deep and detailed understanding of these applications when you go through some of the associated resources that is listed in Introduction to Bioinformatics, and sometimes bytes and molecules in the resources. So with that, I will conclude today's lecture, and in the next following sessions, we will focus more about the important part of these studies that are related to our ability to compare the DNA code from given individual to a reference. Right? This comparison is actually made possible through the computational advances that will take millions of these short reads and align themselves to the reference to extract meaningful insights, as we have been discussing so far. So the next session is to understand the basic concepts of sequence analysis through performing the sequence analysis in an environment like R. So you will start with understanding what is a DNA sequence string and why we need to compare it with the reference sequence, and what do we gain by comparing it with the reference sequence, and what are the variants that we can actually characterize. You can perform this alignment yourself, and what are the different types of alignment that one can focus on, and how to do a test alignment in R's case, and how to do similar analysis by uploading files in our Taubot Bioinfo platform and visualize the output in JBrowser or IGV, etc., etc. That will be our discussion for the next session. And if you have any questions, please let me know in the chat box. And if there are no questions, I will pass on the session to Sparsh.
Yes. So thank you, Dr. Raghav, for this wonderful session. So I'll quickly navigate you all through the program page once again for those who have joined the first pre-introductory session for the very first time. So let me share my screen. Shrikori, could you please confirm if my screen is visible? Yes, it is visible. Okay. So this is the program page, and Shrikori has already shared the link in the chat box itself. So as the program has commenced today from 19th September, and when I scroll down, you can see there is a program overview, which will give you a brief about what, why this particular program is designed, why we are conducting this particular program, and what all benefits one would gain or have if they join this program. Now, on the right-hand side, you can see the interactive sessions labels. The highlighted dates are the dates on which we will be conducting our sessions for Genomic Data Analysis program. So, Kruti, as you asked, the second session is on 23rd of September, that is Friday this week. And today we covered these all topics. So, it was an overview of genomic data analysis program where we covered about the overview of mutations, variations, then molecular determinants of phenotype, course structure, expectations, etc. And the upcoming session is on 23rd, and the title of the session is Unraveling Genomics and Working with DNA Sequences Using R. And here are the topics which we'll cover: understanding of DNA variation, DNA sequences and alignment in R, NGS processing, data formats and technical challenges, mapping, read alignment and section of variants, next steps for whole exome data analysis, working with DNA sequences in R, pairwise sequence alignment, multiple sequence alignment, RNA and protein sequences, exploring PDB, genomic variations, working with DNA sequences in R using toOmicsLogic code block and Google Colab notebook. And here are the associated online resources. Those who are have enrolled for this program for either 45 days, 60 days or 90 days tenure can also see the session recording of the first session, that is of today's session. By tomorrow we upload the YouTube link here on the program page itself. Those who haven't enrolled for this particular program, I will quickly guide you through how to enroll for this program and what all tenures we have for which you can enroll. So as you can scroll down, you can also see the further curriculum of this program, on which particular date the next sessions we will be conducting, their topics, their online associated resources, etc. Now talking about for those particular participants who have not enrolled for the programs but are interested to do so. So this particular program, it is divided into 3 tenures, either 45 days or 60 days or 90 days. Now there are some few common resources which one would avail if they enroll for this program, that are online tutorials, project examples, coding session recordings, online mentor support via Skype, Telegram or WhatsApp groups, online technical support, certificate after completion of the program, and example cloud pipelines on tBioInfo server, which is used for the analysis part. Now, what's the difference between the three? In 45 days tenure, you will have the access to an extensive training related on genomic data analysis and topics or the sessions, and you will have access to online mentor support via either Skype group or Telegram group, or whenever there will be live Q&A session or live session conducted. In 60 days, you have an opportunity to work. You have an opportunity to work or draft a research proposal and present it to our mentor for further evaluation. And then also you can conduct a one-on-one mentor sessions so that you can get your queries clarified via one-on-one Zoom meetings or so. And in 90 days, you have an opportunity to work on an independent research project, and further the outcome is a research publication. And also one-on-one mentor sessions is there. Also, you have an opportunity to get a certificate of excellence after you will complete your research project via research fellowship program under us in 90 days. Now, talking about the pricing, for 45 days, it's 7,500. For 60 days, it's 10,000. And for 90 days, it's 15,000. And right now, we are providing with scholarships, and I put in the chat box. So Shri Gauri has already shared all the details. So my email ID is there. That is marketing@toOmicsLogic.com, on which you can mail regarding if you want any scholarship or so. Also, there is a contact number which he has shared, that is 9876134120, and it is active on WhatsApp. So whomsoever across the globe is active on WhatsApp and they need to directly have a one-on-one conversation with me via WhatsApp call also or a Zoom meeting, they can ping me on the WhatsApp number, or even they can mail me and we can fix exploratory meet to discuss further and what would be the possible discounts on the scholarship one could have. So this was all about the registration part and the scholarship. And yes, we will be providing with the insights of the session 1 for sure. And those who haven't enrolled for this particular program, please enroll yourself up till the next
session, that is on September 23rd, Friday. So please let me know if anyone is having any query related to the subscription part or registration part or the fee or scholarship. I'll be happy to answer. Thank you.
All right. With that, I hope each of you were able to learn something new from today's session. And like Sparsh said, if you have any queries regarding the program details or registration or fee structure, please feel free to reach out to her via the email or the contact number that I've provided in the chat box. And with that, thank you all for your time and patience. I hope to see you all for session 2 of the program. With that, thank you all once again and have a great day. Thank you, everyone.
Thank you, Sri Gauri, and thank you, everyone.